see it?
Yes.
Okay,
Yes.
thank you. So thank you all for being here. And in this opportunity, I'm going to talk about the origin and early evolution of turtles, trying to identify the main problems and the possible solutions from a cladistic perspective. I guess that everybody knows that turtles are amniotes that are characterized by the presence of a shell. This shell is around the thoracic region in the body, and the shell is composed by bony plates that are covered by scutes. This shell, of course, had some impact in different systems in the organism. For example, they have impact in how the turtles move, how they reproduce, how they breathe, and in many other aspects. And maybe as well, the presence of the shell causes the obscure, for example, when we try to understand the homology of this structure with other parts of the body of other amniotes. So nowadays, turtles are a moderate, diverse group of amniotes. They are more or less 357 living species. They have a long history in the fossil record. The oldest turtles are from the late Triassic, 210 million years ago, approximately. They live and they have lived in all oceans and continents, except Antarctica. And they have conquered different environments like the terrestrial, marine, and freshwater. Also, they have a diverse diet. Some are herbivores, some piscivores, and omnivores. But I'm not going to speak up in detail about the biology of turtles. I'm going to focus on the phylogeny of this group, mainly in two main questions. So what do we know about the origin and evolution of turtles? Those are my two main questions that I would like to talk about today. What about the early evolution of turtles? In the last 15 years, we have a global matrix about the evolution of turtles. This global matrix is mainly morphological, and it was started by Walter Joyce in 2007. And since then, we have been working on this matrix, adding new characters, new taxa, and nowadays we have around 100 taxa and 300 characters. When we analyze this matrix using cladistics, we might have this as a result. I'm not going to start speaking about this phylogeny in detail. I'm just going to summarize it in this. So we have a crown group, turtles, that we call it testudines, that is formed by two main clades, the pleurodires, that are those turtles that retract their necks inside the shell in a horizontal plane. And then we have the other clade, the cryptodires, that are those turtles that retract their neck in a vertical plane inside the shell. In the stem of the testudines, we have a bunch of extinct taxa that are located in the stem, and the oldest representatives are from the late Triassic, here represented by Proganochelys. So all this total group recognized as testudinata. The main synapomorphy of this clade is the shell, as I was mentioning before. So we have information about the evolution of turtles in using morphology, but as we have also living representatives of turtles, there are other phylogenies based mainly in molecular data, as the one we have here is a large molecular phylogeny of 80% of the living species of turtles, where we can find a similar arrangement. That we have the crown group testudines formed by two clades, Pleudorida, Pleurodira and Cryptodira. But what happens if we compare this molecular phylogeny with the morphological phylogeny? We have some clades that are similar, but then we have several differences. I'm going to focus on the main, perhaps the main difference between the two phylogenies, that are the relationships of the main clades of Cryptodira, all these clades. When we try to combine the molecular set of data with the morphological set of data in a total evidence analysis, we unfortunately get some unorthodox results, as the one we can see here. So we have again the Studines here present, the crown group turtles, but as you can see here, the Pleurodirs are nested inside the Cryptodirs, that they are Cryptodirs then are no longer a monophyletic group. So we were like wondering why this was happening. So if we compare the unrooted tree of the main clades of Cryptodirs, that are these main clades in here, these 5 clades, we see that in general, the relationships between these clades in unrooted trees are similar in the molecular and in the morphological analysis. However, the position of Pleurodirs are different in both analyses. In the molecular analysis, Pleurodirs are more closely related with Trionychia, while in the morphological analysis, they are more closely related with Chelonioidea. And when we combine both sets of data in a total evidence analysis, we get the topology of the molecular analysis, but with the root from the morphology. And that's why we get this paraphyletic Cryptodir. We didn't give up, and after some years of these studies, we were trying to do again total evidence analysis with larger matrices, both coming from the molecular set of data with more turtles, with more genes, and also from the morphological point of view with updated matrices. But again, the results were unorthodox. As we can see here, these are not published data. These are analyses that we are working on with Danieli Pereira. And in here you can see this the total evidence analysis, where we can see in black are the clades that are in agreement with previous phylogenies, but in the base of the tree we can see that with red these weird clades not found before. So we are trying to understand why this is happening and trying to use different strategies to get maybe better results. So what about the other question that I was posting today about the origin of turtles? Maybe that's one of the most controversial thing about the amniote evolution, because we don't know exactly which is the position of turtles in the amniote tree. Maybe because it is obscured by the presence of a shell, but it is certain that turtles have been related with mainly all clades of amniotes. As you can see here, this is not exaggerated. On the contrary, I was trying to post the main hypothesis. So if we see this this simplified tree about amniote evolution, we see that turtles have been related with diapsid reptiles as crocodiles, birds, also as sister groups of archosaurs, sister to lepidosaurs, to extinct marine reptiles, and also as the sister group of all diapsids. Also, turtles have been related with captorinids, that as the formed a sister group with of Diapsida, and also turtles were related with some anapsids or parareptiles like pareiasaurs, procolophonids, and eunotosaurs. Mainly those hypotheses coming from the molecules, from the state of data coming from molecules, relate turtles with crocodiles, birds, and lepidosaurs. While those phylogenies using morphology relate turtles with anapsids and also with diapsids as well. So there are many phylogenies. I'm not going to go into detail about this, just to summarize that there are many phylogenies since the 90s until recently that relate turtles with diapsids. Also, there are other phylogenies that recover turtles as the sister clade of all living diapsids. From the morphological point of view, we have that three main hypotheses. Turtles related with captorinids as the sister group of diapsids. And this was proposed by Gauthier et al. In 1988, and was one of the first cladistic phylogenies published about the amniote evolution. And in this paper, they highlighted the importance of including extinct taxa into phylogenies to try to understand the interrelationship of taxa. So among the morphological hypotheses, we have several papers supporting these relationships. In this case, the anapsid skull of turtles would be a synapomorphy for the group, while if we consider turtles inside this parareptile clade or anapsid hypothesis, we will see that the anapsid condition in the skull of turtles would be plesiomorphic. There are a bunch of papers also supporting this, the affinities of turtles with parareptiles. So why we get such an array of different hypotheses? So we wanted to look further into this because and I believe what is happening is this. It reminds me of the Indian parable of the 6 blind men and an elephant, where each man is touching a different part of the animal, and it's saying, okay, I have this is the tail. Oh, so this is a rope. And the other man is touching a leg and says, oh, this is a tree. And the other man is touching the ear, says, oh, this is a curtain. But no one is speaking to each other and put all the information in a general context. So I think this is what is happening with the origin of turtles. It's like each part of each researcher or group of researchers are looking at a certain part of the animal, either the molecules or the morphology, but we need to understand this in a more total evidence framework. So if we analyze all at least the main papers about the origin of turtles, we can see that 50% is based on morphological phylogenetic analysis, where maybe the character sampling is not so big. And then we have around 42% of the papers coming from the analysis of molecular phylogenies with few genes and few proteins. But okay, there are exceptions, some papers with thousands of them. And then only around the 8% is in a total evidence framework. So it's very little compared with all the remaining papers. And if we see the analysis, the morphological analysis, 60% of them are relating turtles with diapsids, 35% with anapsids, and 60% are sister to diapsida. And the molecular are all related, mostly related with diapsida, and only 13% relates turtles as the sister to diapsida. The total evidence analysis, most of them have found turtles inside diapsida, and only one of them found turtles as sister to diapsida, as is Golovov's et al. 2009 paper. So if we want to analyze this, so why is it happening? Why we get these results? So we were trying to look into the matrices of this analysis, and we found that there is a biased taxon sampling. For example, in the anapsid hypothesis, in general, 3 out of 5, there are more anapsids than diapsids included in the sampling. And when I say more, it's way more, yeah. And in the diapsid hypothesis, as a rule, 15 out of 16, there are more diapsids than anapsids. And some of the analysis did not include anapsids at all. And those papers where the result is turtle sister to diapsida, no anapsids are included in, this is 3 out of 3. So really we have a biased taxon sampling in those main analyses. There is also a poor taxon sampling. Analyses are in general with less than 80 taxa, except Golovov et al. 2009. And in general, they are about 40 taxa. So they are analysis with a little taxon sampling. Of course, if we have a biased taxon sampling, we have biased character sampling. And also we have found that there are contradictory coding of the same taxon in different analyses. So that's not so good. And of course, in this total evidence analysis, there is a lack of anapsid representatives. So I think we are in this situation, and that's a problem because all those phylogenies are used, and new studies are coming and are based on those phylogenies. So we would like to propose some changes to this, for example, to include taxa from all the clades that have been suggested as sister groups, because all this seems really obvious to when I was writing this, but it seems they are not so obvious because we keep on doing the same mistakes. Include as many taxa as possible, and taxa at the species level, not higher, because again, many of these phylogenies I was showing to you, they don't, the terminal taxa are not species, but they are higher groups like families or even higher. So maybe they are just scoring chimeras. If more tags are included, more characters should be included as well. And of course, we need to check the codings firsthand because, as I was saying before, there are some contradictory scorings. And we need not only to include morphology or molecules, but we need to do it more in a total evidence analysis, in a total evidence approach. So this is what we are trying to do right now. Together with Evan Blajos, we are working on a super matrix, trying to gather as much information as we can. This is we started with a morphological matrix, where we are gathering the latest versions of the matrices about synapsids, parareptiles, archosauriomorphs, turtles, of course, and lepidosauriomorphs, to include it in a super matrix. Nowadays we have 2,200 morphological characters and more than 330 tags. But we are still far away from the end. We need to do, for example, we need to check all the characters, because there are duplicate characters, there are characters that are not quite similar between different phylogenies, characters with more states in one phylogeny than in another. So we need to do that first. Then we need to check the codings of all the taxa, and we need also to gather the information from the molecular data set. And after that, we need also to cross our fingers to get a result of this and not a complete polytomy. So unfortunately, I don't have results for this part of the talk. It was just to raise this awareness about the origin of turtles, because it's a very debatable subject in amniote evolution, and nowadays it seems that this is solved, that turtles are diapsid reptiles, but I think we are not there yet. So I hope we will not fall into this again, and not to be blind in this respect. So to summarize this talk, I was speaking about the molecular phylogenies of turtles, the morphological phylogenies, and the problems between brackets that appear or that we need to face when we combine both set of data. And also I was talking about the different hypotheses about the origin of turtles, and I was trying to identify also the problems that we face when we try to understand the phylogenetic position of this clade among other amniotes. And I was presenting you the first steps towards a more inclusive phylogeny about the origin of turtles. So thank you very much to all of you, and Christian and Dolores, for the invitation to give this talk.